Welcome to Halteres Presents, a brand agnostic interview podcast that seeks to objectively highlight the happenings within the world of diagnostics. And now, your hosts, Thayer and Mickey Urdea.
So hello and welcome to Halteres Presents. My name is Rich Thayer, Managing Partner at Halteres Associates.
And this is Mickey Urdea, Founding Partner at Halteres Associates.
Today we are joined by Sir Michael Houghton, Nobel Laureate in Medicine and Director and Professor on the Faculty of Medicine and Dentistry at the University of Alberta. Michael, with co-inventors Quilin Chu and George Kuo at the Chiron Corporation in California, and in collaboration with Dan Bradley of the CDC, discovered hepatitis C virus and first published their findings in 1989.
We go a long way back with Mike. We were together at Chiron Corporation. We were there at the time when his team was discovering the hepatitis C virus, and Rich was a key part of that. So we're very glad to have been able to catch up with Mike and had a great conversation with him about what's been going on in the world of hepatitis C since all those years ago, including his own current work in vaccines.
Yes, Mickey, and towards the end of our interview, Mike had some wonderful thoughts for the next generation of scientists and opportunities in molecular medicine and testing. And now our interview with Nobel Laureate, Dr. Michael Houghton. Welcome, Mike. Please tell our audience a bit about yourself and your career.
Yes, hi, Rich and Mickey. Great to see you again after all these years.
Great to see you.
Thank you. Currently, I work at the University of Alberta in Canada. I went there in 2010 to take up a chair, which had a very nice grant attached to it. And I'm working, believe it or not, on a hepatitis C vaccine, which is kind of the missing link right now in the hepatitis C story. But also we work on other viral vaccines, and also we're branching out and we're working on a new approach to Alzheimer's therapy. So I direct a translational institute at the University of Alberta. And I eventually left Chiron in 2007 after working there for 25 years. And at Chiron, my lab with colleagues and collaborators discovered the hepatitis C virus in 1987, and we published in 1989.
Thank you, Mike, for that introduction. The road from discovery to cure for hepatitis C has been an amazing odyssey. I remember working with you as part of the HCV discovery team at Chiron Corporation back in the mid-1980s when you and your team discovered the hepatitis C virus. Of course, back then it was known as non-A, non-B hepatitis. And now you and Doctors Harvey Alter and Charles Rice were awarded the Nobel Prize in Medicine in 2021 for your efforts in discovering and culturing the virus. Please relate for our audience what it was like back then, working to discover Hep C, and what it was like on that Saturday morning when it seemed like it might have happened.
Well, at the time, it was quite a lot of pressure, actually, because, I was working for Chiron, which was a startup. I joined them in 1982, and it had been going for about a year at that point. But like all startups, money was scarce, and the conditions of the lab were interesting, shall we say. My lab had a leak in the roof whenever it rained, because, of course, financing is usually not great to begin with in a startup. So most of the work people at Chiron were doing was contract research. I think Mickey had his own program on branch DNA, but most of the programs were sort of funded by external organizations. But we decided to see if we could discover the cause of this important hepatitis agent, then called non-A. It wasn't Hep A, it wasn't Hep B, but nobody really knew what it was, so people called it non-A, non-B. So we thought with the molecular biological techniques that I had experience in, and Chiron in general, we thought maybe we could crack that nut and go on to develop a lot of products following that discovery. So I think it was very memory inspiring, for sure. It was a great time. Had a lot of great colleagues that I really enjoyed. But there was some pressure as well. You know, it was always, it took at least 5 years of pretty intensive research before we found the cause of non-A, non-B and called it hepatitis C. So there was a fair bit of pressure, but great times also.
So, Mike, thanks to the efforts in discovering the virus, you and your colleagues at Chiron, and the efforts of folks at, for instance, sharing Plow early on with interferon treatment, and later, of course, Gilead and others to develop and launch these direct-acting antimicrobial treatments for Hep C, the disease is now curable, which is astonishing.
Yes.
This is a remarkable accomplishment in medicine and could directly lead to saving millions of lives around the world that just couldn't have been saved previously. So please share with us a bit about your observations about the path from discovery to cure. And, for instance, how did what we've learned about the biology of HCV contribute to improved treatment over time?
Yeah, thanks, Mickey. Well, at the time, I was a little frustrated that it took so long to develop antivirals, specific antivirals against Hep C following our discovery of the virus. We discovered, we first published our findings in 1989. And so I thought, over the next 10 years, we would have antivirals out on the market. But it actually took 25 years before Gilead came out with the first direct-acting antivirals. So, my lab at Chiron's, we participated in that general area of antiviral discovery and development. We identified some of the key enzymes involved in the replication cycle of Hep C, which obviously became drug targets. And because we characterized the virus, its proteins, its enzymes, and of course its genome, I believe Chiron out-licensed those patents to the drug companies around the world. So it was nice to have contributed to the antiviral development. I was very impressed with the work from Gilead. They really did a wonderful job bringing out their cocktail of antiviral molecules that could now cure nearly all patients infected with Hep C, irrespective of the genome that they had. There's multiple heterogeneous forms of Hep C. And so that was fantastic and so important for the patients, because hepatitis C, it generally develops as a disease. It develops quite slowly in most people. But in some, a subpopulation, liver cirrhosis develops quite quickly. And that can be a severe clinical disease. And then from cirrhosis, it can develop into liver cancer. And so being able to cure patients was a fantastic milestone. And not only cure them, but cure them quite quickly. Just in most patients, 2 to 3 months of oral therapy that was well tolerated cures them for life. So yeah, it was really wonderful to see that. And of course, after our discovery at Chiron, we initially focused on developing blood tests to protect the blood supply and to diagnose patients to help their clinicians in treating their patients. And you were involved in that, Mickey, with the BDNA discovery and development. Once we did that, then at Chiron, we focused more on the vaccine side of things. And as I mentioned, we out-licensed our patents to different companies around the world. It took a long time. I was surprised. I think, the protease was the first target for antiviral companies. And the problem with the Hep C protease is it doesn't have a very well-defined tertiary structure. So it's not so easy to get drugs in at the active site of the protease. And that means they had to make quite large molecules interacting at different points. And when you have to do that, you can get some problems with solubility and affinity and also toxicity. So I think that's really why it took so long. But it's great to have developed the blood test to stop people getting it after a blood transfusion or after receiving blood products. And great now that we have good antivirals. And what we lack is a good hepatitis C vaccine. So I spent a lot of time at the University of Alberta developing an improved version of the Hep C vaccine, and hopefully we'll be in the clinic very soon with that.
I remember well, you were involved with vaccine development so early on, and here we are all these years later. What have been some of the issues with developing a vaccine for HIV, and are things such as mRNA vaccines that have been so successful with SARS-CoV-2 now going to play a role with HCV, you think? So where's it all going?
Well, I think you're right, Mickey. The RNA technology has emerged shortly after the COVID pandemic struck, and certainly it is being applied to Hep C protection, and my lab is working in that area also. But I think to answer your initial question, in developing a Hep C vaccine going back to the 1990s, the first challenge was it was very difficult to grow that virus in cell culture. And without doing that, it was hard to develop an assay for neutralizing antibodies. So nearly all vaccines approved for different viruses and different bacteria, they rely on antibodies to neutralize the infectivity of those pathogens. And of course, if you can't grow the virus in cell culture, it's very difficult to develop a neutralizing antibody assay. So that was the first problem. And it wasn't until the early part of this millennium that people, actually a Japanese researcher, figured out a way with a particular strain of Hep C to grow it in culture. And also a group in the Rockefeller was able to identify the production of pseudoparticles, not actual virus growing in cell culture, but pseudoparticles. And those tools enabled us to start actually quantifying neutralizing antibodies. So once we had that, it became easier to develop a vaccine. And I think the other problem with vaccine studies on Hep C is that we didn't really have a convenient animal model without having to use chimpanzees. You know, there was no small animal model. And so chimpanzees are endangered species. They're now not available for research in the United States and worldwide. We did use them in the early part of the vaccine program, but I think lack of a convenient, easy animal model has been a big problem in developing vaccine. And then you start to develop a vaccine and you start making headway, you start getting into phase 1 trials. And then, of course, when the antivirals developed and were approved, a lot of people said that, well, now we don't need a vaccine. And so I think that actually stunted the field of Hep C vaccination quite a bit. This incorrect conclusion that we don't need one. Why is it incorrect? It's incorrect because if you look at the history of infectious disease, you have to have a vaccine to prevent the epidemic and prevent transmission within the community. Drugs alone don't do that. If you look at things like syphilis, gonorrhea, chlamydia, these are sexually transmissible pathogens. Using them as an example, we've had cheap medicines to cure people of those, antibiotics, and yet we still get outbreaks of those sexually transmissible diseases. So it's an example of why you need vaccines, and smallpox would not have been eradicated from the planet without a vaccine. And as we saw with COVID, while there were some antivirals available, you really need a vaccine. So I think the field took a blow in people thinking we didn't need a vaccine. The status today is that with the opioid epidemic, more and more people are using opioids and they share needles. And so, if anything, the incidence of Hep C is actually increasing around the world. So it's even more important we have a vaccine. So those are some of the issues that I think held back the research, made it difficult and slow. But I do believe now we understand better what are the immune responses associated with protection against hepatitis C. And now we have tools, including RNA, but also more conventional techniques to recapitulate those to develop a vaccine. So we're on a path, I hope, where we can prove efficacy in humans with our vaccine in 2 to 3 years' time.
That's wonderful.
Yeah.
So, Mike, you may be aware that in Egypt, they have a program now that includes the hepatitis C diagnostics, as well as a 12-week regimen for the DAA for well under $100 that they've deployed across the country for testing and essentially eradication of the disease. Since you were awarded the Nobel Prize in 2020, has that increased awareness in hepatitis C translated to similar programs that you're aware of that may be occurring in the United States?
Well, I think the award of the Nobel to 3 scientists did, I think, put more focus on hepatitis C itself. In 2020, when the award was given, we already had good blood tests, good diagnostics, and good antivirals. And so I think it also became aware by that time that there was a growing incidence of hepatitis C around the world. So how can this be? And unfortunately, because of the opioid use and the growing opioid epidemic, and when people share needles, injection needles, it's a very efficient way to spread hep C. I think it did help to make people more aware that we do urgently need a vaccine to control this pandemic. So I think the Nobel helped in making that awareness of the need of a hep C vaccine, gave it a priority, I think. And there are many different groups now around the world working on a hep C vaccine. So it's good to see it in a healthy, competitive state. People are using RNA. We are using RNA as well as recombinant proteins, and people are using different methods, some of them similar to the COVID vaccine discovery research. So yeah, I think Nobel did help to put more focus on hep C.
We have all these other sources of infection too. For instance, in Egypt with the schistosomiasis treatment that led to a very large number of cases and people getting injections of so-called vitamins or other things for their well-being, supposedly, but contaminated needles then as well. So those are much more difficult people to identify. But being able to vaccinate these populations will certainly make an enormous difference.
Yes.
Couldn't agree with you more.
Well, it's a good topic you brought up, Mickey, because as you know, in Egypt, they suffer from probably the worst aspects of hepatitis C in the world, because when they prevented schistosomiasis in their population, unfortunately, they did not use sterile needles between individuals. So unfortunately, hep C really spread like a wildfire in Egypt. It's very interesting, though, how they've dealt with the testing and treatment of their population. So Egypt has now pretty much tested everyone in their country. This is like 70 million people for hep C, and they found 4 to 5 million carriers of hepatitis C, and now they've treated them with the new antivirals and cured them. So that is one way that you can actually eradicate hep C transmission without a vaccine. The way that they did it is the cost of the antivirals for every patient was only $80, I believe, per patient. Now, in the US and other developed, highly developed countries of the world, those same drugs cost $30,000 per patient. And so the pharmacoeconomics actually prevents, for example, the USA doing the same thing. Of course, USA is a bigger population, 330 million or so. To go in and test all of those people for hep C is a daunting task. And then to provide the expensive drugs at $30,000 per pop, becomes a huge burden to the healthcare system. And that is why Egypt has cured Hep C in their country and prevented transmission, but the US hasn't. Dr. Collins, who used to be head of the NIH, he's now got a bill in front of Congress to do exactly what I just described, to provide money to go and test all adults for Hep C. And once the carriers are identified, treat them with the antivirals that can cure them within 2 to 3 months. So that is in Congress. We'll have to wait and see if that passes. But if it does, and it's really performed efficiently, then that could also be a way to prevent HCV transmission without a vaccine. But even if that is so, and it's by no means certain that that will actually get passed, there's many other countries of the world where that's not going to happen and where a vaccine is needed. So, I think we definitely need a Hep C. And, by the way, I will add that a vaccine really can be not just cost effective, it can be cost savings to the healthcare budgets. So rather than diagnose and treat people, which is important to do for patients' health, of course, but in terms of preventing transmission, vaccines are a relatively low-cost way to do that. And going forward, developing a vaccine for, say, people who inject drugs who will get infected in the future will be very cost savings to the healthcare budget. So we need to get the antivirals out to the patients carrying the virus, but secondly, we need to make a vaccine as well.
Yeah, I'm glad you mentioned that, Mike. Interestingly, on a prior podcast episode, we did make mention of the Biden administration's Eradicate Hep C by 2030 program. And, as with you, we're hoping that that bill actually makes it through Congress and gets funded.
Yes.
Yeah.
And okay, so they've treated everyone in Egypt. That doesn't mean they've eliminated all potential sources of new infections. You still need to vaccinate people. So even if we go through this program in the United States, you'll still need a vaccine.
Yes, I think so, Mickey. Egypt is a diverse population. A lot of people come in from other countries into Egypt. And like I mentioned, most countries are not able to do what Egypt did, test and treat all their populations. So I think Hep C vaccine is going to really eliminate hepatitis C as a major global threat. So still working on it. Chiron and then Novartis had a program on it, which I worked on, and it was going well. But unfortunately, after I left Chiron, Novartis, as it then was in 2007, after I left, Novartis focused their resources on meningococcal disease as well as influenza, and did not go through with a efficacy trial of my vaccine, which was the intention when I left in 2007. So, I understand how priorities change in companies, but it was disappointing to me, obviously, having worked on the vaccine for many years, that Novartis never went and performed what was going to be a big efficacy trial in the United States. I think if they had done, it had ingredients in that vaccine that I think would have been partially protective, and it would have helped. It would have curtailed the incidence of Hep C infection around the world. Yeah. However, my work in Canada, we've improved upon that vaccine in many ways. So I'm still optimistic we can produce a vaccine. So that's what I'm working towards.
Fingers crossed.
Good luck with that. Mike, I can't remember what year it was, but it must have been in the mid-90s. You and I were scheduled to give a talk in Spain at the same time. I was talking about viral load testing and you were talking about a vaccine. And you called me like the night before the meeting and said you were not going to be able to make it. And you asked me to present your vaccine paper. And you sent me the papers and in the slides. And I studied like crazy all night to go before that august group of people who knew all about this and present your work for you. So that was quite interesting.
Well, I had forgotten that, Mickey, and thank you so much for doing that.
This episode of Halteres Presents is brought to you by the UC San Diego Extended Studies In Vitro Diagnostics Product Development course. Are you looking to take your biotech career to the next level? The UC San Diego Extended Studies In Vitro Diagnostics Product Development course is a unique, live online program designed for managers, entrepreneurs, investors, and scientists. This team-taught course by award-winning industry experts covers everything there is to know about the requirements for moving an IVD product from concept to regulatory approval and market launch. Explore real-world product development cases to help you plan for success by covering topics such as setting product requirements and specifications to meet customer needs, critical path management, resource planning, principles of assay development, instrumentation, role and composition of the product development team, risk analysis, and IVD clinical and regulatory issues such as compliance and complaint handling. Ready to get started? Enroll today and use our exclusive discount code to get a 10% discount on your enrollment. Use code HALTERES10 at checkout. That's HALTERES10 for 10% off the price of your UC San Diego Extended Studies In Vitro Diagnostics Product Development course. Just check the link in the show notes for this episode and use code HALTERES10 to enroll today. Accelerate your in vitro diagnostics career with the UC San Diego Extended Studies program. Your future in biotech starts here.
By the way, going back to your earlier question on why the vaccine for Hep C has been slow in developing, another reason was that very, very well-known institutions, who I won't name, actually really were very pessimistic in their publications. They were saying, well, Hep C is more heterogeneous than even HIV. And we all know the problems of heterogeneity in developing an HIV vaccine. So Hep C vaccination was going to be impossible. And I always thought at the time, well, really that's unnecessarily exaggerating the situation. And, you know, today I can tell you that despite that heterogeneity of Hep C, within the viral envelope glycoproteins, there are two. It turns out there are highly conserved antibody binding epitopes, which when the antibodies bind, they do neutralize the infectivity, not only of that strain, but of most strains that we see occurring around the world. So I think, you know, science does have to be careful about jumping on the bandwagon. And there is a certain fashion that occurs in science. And it was a little frustrating to me as a vaccinologist working on this problem to see such pessimistic predictions about the difficulty of making a Hep C vaccine. From the point of view of heterogeneity, those fears turned out to be unfounded, I believe.
I remember back when all the subtypes and genotypes were being discovered, and I don't think we really realized at the time how different they were going to contribute to the use of antivirals until fairly recently. And I just remember looking recently at one of the guidelines for hepatitis C treatment from about 2015 with all the different recommendations for type 1A versus 1B or genotype 2. They were all different combinations. And it's just so wonderful that's now all coalesced into one treatment for everyone with Hep C, independent of what their genotype is. That is just phenomenal.
Yes. Yeah, I agree, Mickey. I think there was a little too much pessimism about developing antivirals for all the different isolates of Hep C. I raised it because I think in the vaccine arena, it did actually result in underfunding of the vaccine. You know, the NIH has spent some money on HCV vaccine, but nothing like as much on other infectious pathogens. So, you know, I do think when scientists publish their papers, they need to be a bit careful about extrapolating their data to future developments, because it can affect funders like NIH, like CDC, and also private capital. You know, if a private investor is reading a paper from an august researcher and he or she is saying, well, there's no chance of developing a Hep C vaccine because it's too heterogeneous, you're not going to spend your money in a program to do that. And in fact, sort of that's what happened with Novartis, I believe. They felt it was a safer proposition to develop flu vaccines and meningococcal vaccines than HCV.
Yeah, that's very interesting. Mike, you were talking a bit about some of the problems with developing vaccines, and we were talking a bit about that with antivirals as well. And I just wondered if you wanted to expand a little bit about this issue of cultured virus as well in the antivirals, because we had the luck of being able to culture HIV, and we could culture HBV and a number of other things, but not HCV. That was an impediment to the antivirals as well, right?
Yes, it was, Mickey. Fortunately, once again, it took time, but two excellent virology researchers developed what's known as the Hep C replicons. So these were subgenomic RNAs that after a lot of effort could be shown to replicate in mammalian cell cultures. So one of the groups was Charlie Rice, who shared the Nobel with me, and another one was Ralph Bardenslager in Germany. They published about the same time. I think it was around early 2000s. And what those replicons did is everyone developing antivirals to Hep C used them to test the potency of their drug candidates. And so that was a very helpful point in drug discovery and development for Hep C. And then afterwards, a Japanese researcher found a particular strain of Hep C that could replicate the whole genome and produce virus. Dr. Wakita developed the first method for replicating the whole virus in cell culture and producing infectious virus. It was using one particular strain, it was a genotype 2A strain, and really that was a breakthrough because now drug developers could test their drugs for inhibiting the whole life cycle of the virus, and then also vaccinologists could determine if their antibodies can inhibit the infection and the life cycle process. So it was a key development. That did not happen until getting on for 20 years ago, so early this millennium.
Is that something that researchers in general have access to now?
Well, yeah, unfortunately, only one strain can actually replicate efficiently in cell culture. But Jen's book in Copenhagen, in Denmark, what he did is to actually take that one strain, and then he would take out the envelope glycoproteins of that strain and put in envelope glycoproteins from all the other strains. And so that clever trick enabled us to be able to assay for broadly cross-neutralizing antibodies to all the viruses displaying their own envelope glycoproteins, because most neutralizing antibodies bind to the surface proteins in the envelope of the virus. And so that clever trick helped us a lot, and we used that method. In fact, we used that method in 2013 when we published a paper from University of Alberta to show that humans immunized with the two envelope glycoproteins derived from a single strain of Hep C could actually cross-neutralize many of the global genotypes of Hep C. So it was the first demonstration that clearly there are highly conserved epitopes which antibodies bind to, and then neutralize the infectivity of not just that strain, but most of the other strains.
That's certainly exciting. Mike, what do you think are the most exciting advances that have occurred in molecular biology and medical science since you discovered hepatitis C that you believe may lead to a much quicker timeline from discovery to cure for other critical diseases?
Yes. Well, that's a great question. And, when I look back at the technology in the 80s, when we were trying to find Hep C, it took us getting on for 7 years to discover it, the virus, and then to characterize it. As we saw in the case of COVID, it took months. not even 7 months, closer to 7 weeks. Why? Because the technologies that were developed between when I worked on Hep C discovery and today, incredible technologies. You know, back in the 80s, PCR, polymerase chain reaction amplification of nucleic acids, was not really routine use until the end of the 80s. Really, after we had discovered the virus. If we had had PCR in the early 80s, I think we would have found it faster. And then deep sequencing of the genomes. Yeah, Rich, I remember you sequencing our first clones of Hep C and me looking over your shoulder. And I think you will probably recall it took several weeks just to get a couple of hundred nucleotides of sequence.
Absolutely.
Now, the entire human genome can be sequenced in a few weeks. So that has really obviously enhanced infectious disease and all aspects of medical research. I think now we have computational drug discovery to really aid the identification of new antiviral hit drugs that can be developed into approved products. So it's just amazing. It's one thing that we've emphasized up at the University of Alberta. It will take a pharmaceutical company many months in a wet lab to screen 500,000 drug compounds, you know, usually a cell-based assay or a biochemical assay. And they're very good at that. And it obviously works. But now in the computer, in supercomputers, you can screen billions of drug fragments in silico. And what that does, it actually accelerates drug discovery a lot. So we have those techniques that most groups around the world are using. Now we have gene editing techniques like CRISPR, which you can knock out genes and you can knock in genes. So you can identify, obviously, key processes of disease faster. So yeah, when I give lectures to students, I usually tell them it took us 7 years to discover and characterize Hep C, whereas now it will take 7 weeks. So it's just wonderful. And by the way, when I'm giving my talks to students, I tell them this with all sincerity. This is a great time to get involved in medical research and medicine because the technology has so advanced and it's still advancing exponentially. This century is going to be the golden age of molecular medicine, I believe. And there's so many diseases out there about which we know very little, let alone having drugs and vaccines to prevent them. But I think by the end of this century, and probably well before, most diseases are going to be well controlled. And I just think for young people entering molecular medicine or medicine itself and related themes, nowadays, it's not just biochemists or molecular biologists or MDs, it's physicists, it's chemists, of course, it's physical chemists, it's computational scientists. It's going to be a wonderful golden age of medical discovery, I believe, this century.
Cross-discipline, yeah.
I think with all of our knowledge around the world, I think we can be doing better than what we are. I mean, when you think of all the dreadful diseases out there, multiple sclerosis, lateral sclerosis, inflammatory bowel disease, Alzheimer's, other neurological problems like Parkinson's, I'm kind of beating myself up and my fellow scientists a little bit here, perhaps unfairly. But we have the ability now to do something about these diseases. And so we need more funding to focus on all these terrible diseases. And also we need scientists willing to focus on disease. You know, a lot of academic scientists, they do a great job in finding new processes of the cell or the virus or the bacterium, which translational scientists can use. But I also think there needs to be more focus on disease medicine. And indeed, the NIH has moved towards that, and I'm pleased to see that.
Yeah. You know, one of the advances that I'm very excited about is this whole issue of spatial biology and looking at small groups of cells within larger groups of cells and understanding their contribution to disease. And I suspect that that's going to help accelerate all this too. And a lot of the things we don't know because small populations of cells that have not previously been identified are the culprits. So, you know, recently I know people have discovered in kidney cancer, adult and pediatric, a small set of previously unknown cells which appear to contribute to the initiation of the cancer process. So, you know, wow, if we start having all that information in addition to what we know now and can really understand the biology at a much deeper level,
Yes.
that's going to explode this whole area, I think.
Yeah.
All these diseases. Yeah.
Very good point, Mickey. Yeah, I think it's a great time to get into molecular medicine. It really is. And cell biology, as you're pointing out. And of course, what we need at the same time is nations to get on with each other and not go to war. Sometimes I feel very frustrated being part of a huge global community fighting disease, and yet every day on our TVs we're reading about wars. We've also got to improve as a species. Let's make everyone healthier and have great lives, but somehow we've got to stop fighting each other.
Sounds like a good plan to me. Need more collaboration across countries. Really make the greatest impact.
I think it's an important discussion, Mickey, because we need the youngsters to get involved in the political process, because really old people really have not come up with the answers.
Yeah.
Probably a sentiment expressed by every generation. Mike, is there anything else in the last minute or two here you'd like another question to address, or any other topics, you know, perhaps pertaining to your research, or anything that you just want to chat about?
Well, yeah, thank you, Rich. I would like to say this, that, you know, the Nobel Prize was given to me along with Charlie Rice at the Rockefeller and Harvey Alter at the NIH. And those two scientists have made huge contributions to Hep C and other areas, and it was very well deserved. But I would like to say that I had two colleagues at Chiron, Quilin Chu, who worked in my lab, and next door, a very good colleague and friend, George Kuo, had his own lab. And really it was the three of us working together that came up with the discovery of hepatitis C. So I really would have liked Quilin and George to have shared the Nobel Prize, but the Nobel Foundation only gives it to a maximum of 3. So, but I would like to acknowledge the critical roles of Quilin and George in the discovery of the virus at Chiron that we first published in 1989. Also, Chiron had a lot of really good people. So, you know, I had lab fulls. I guess I had 2 labs in the late 80s. I have labs full of really good graduates, BScs, MScs, PhDs, who really did a fine job. Really good colleagues like Amy Weiner. That
Amy.
Want to emphasize again how difficult it was to find this virus in the 80s. You know, it really was tough. And Amy tried a lot of things without success, as other people did. Kengsheng Wang and many. I mean, we must have tried 30 or 40 approaches before finding the one successful one. And we have fantastic support at Chiron. And, you know, without that, we certainly wouldn't have characterized the virus as fast as, or as slow as we did, depending on your vantage point. Yeah, so wanted to say that. And also, as you pointed out, Rich, earlier, it is the golden age of molecular medicine that we're in, but interdisciplinary science is absolutely key these days. You know, it's no longer the territory of cell biologists or molecular biologists. You've got to work together with chemists, physicists, MDs, and you've got to work with public policy people. I can't tell you how important it is to have young people going into the public policy arena. We have seen just what happens when you have a great public policy in Egypt to go and test and treat your Hep C carriers in your entire population. So we need all sorts to conquer disease, and I'll end there.
Well said. Mike, this has been a wonderful session with you, reminiscing and thinking about what's transpired since the discovery by you and your team on hepatitis C.
Thank you, guys. Great to see you all.
Thank you so much, Mike.
Thanks so much.
Halteres Presents is produced by Halteres Associates, a US-based bioscience consultancy that provides strategic and tactical services in the areas of diagnostics, medical devices, and life science research to clients of all sizes. The views, information, or opinions expressed during the episode are solely those of the individuals involved, and Halteres Associates is not responsible for any errors or omissions or for the results obtained from the use of this information. The information provided in this episode is for informational or educational purposes only and is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment. Halteres Associates would like to say thank you to this episode's guest or guests, and thank you for listening to this episode of Halteres Presents.